transplanting stool from a healthy donor to treat Parkinson's disease. It sounds like a joke. It isn't. It's a real treatment, and in recent years, several research teams have put it through serious, randomized clinical trials in different countries around the world. A team from Loudi Central Hospital in Hunan, China, led by Dr. Beniao Liang, has just published the most complete analysis so far on whether it actually works. We're going to go through exactly what they found, how they measured it, and what it means for someone living with this disease today. Parkinson's disease is the second most common neurodegenerative disorder in the world, and an increasingly common source of disability and reduced quality of life in older adults. The current standard treatment relies mainly on levodopa and similar drugs, which replace the dopamine the brain stops producing. They work well at first, but effectiveness fades over the years, and motor side effects start to appear. That's exactly why there's so much scientific interest in complementary treatments, approaches that wouldn't replace medication but could act on other mechanisms of the disease, including one that happens far from the brain, in the gut. Typical image of Parkinson's is tremor and stiffness, but the disease is frequently accompanied by gastrointestinal dysfunction too, and it goes well beyond movement. It's often accompanied by a broad range of non-motor symptoms: sleep problems, mood changes, memory issues, and very notably, digestive dysfunction. And that's the key to this whole story. Growing evidence suggests the gut may play an important role in how Parkinson's actually develops. Researchers call it the gut-brain axis, a constant, two-way line of communication between the bacteria living in our gut and the central nervous system. Previous studies had already shown that people with Parkinson's tend to have an altered gut microbiota, with a reduced abundance of beneficial bacteria and a relative enrichment of pro-inflammatory bacterial groups. That imbalance doesn't necessarily stay in the gut. It's been linked to intestinal barrier dysfunction, inflammation, and abnormal neuroimmune signaling that could reach the brain through the immune system. Gut bacteria are also thought to influence the production of short-chain fatty acids, intestinal permeability, and inflammatory signaling, all of which has made restoring microbial balance an increasingly attractive therapeutic target. Among the ways to intervene on the microbiota, fecal microbiota transplantation, FMT for short, is one of the most direct, transferring stool and its bacteria from a healthy donor into a patient. FMT already has proven therapeutic value in several gastrointestinal and metabolic conditions. In Parkinson's, early clinical observations suggested it might reduce constipation and ease certain motor or non-motor symptoms in some patients. But the available clinical evidence stayed limited, and results were often inconsistent from one study to the next, partly because of differences in fecal preparation, delivery route, dosing frequency, and how long patients were followed. Given that emerging but inconclusive evidence, what the field really needed wasn't another isolated study. So instead of running a brand new trial, this team did something more valuable. They systematically reviewed and combined every randomized trial published on the topic so far. That kind of work is called a systematic review and meta-analysis, and when it's done rigorously, it carries more scientific weight than any single trial on its own, no matter how well designed that one trial was. The study followed the PRISMA statement, the international standard for this kind of review, and it was prospectively registered in Prospero, the International Registry for Systematic Reviews, under registration number CRD 420,251,121,443. Registering the protocol before doing the actual work matters. It's a public commitment to the methodology before anyone has seen the results, which closes the door on quietly changing the rules once the data comes in. Search covered 4 major databases: PubMed, Ovid Embus, Scopus, and the Cochrane Central Register of Controlled Trials, from each database's inception through September 15, 2025, plus a manual check of reference lists from every eligible article. Interestingly, clinical trials. Gov itself wasn't searched directly, only trials indexed through those 4 databases. 2 reviewers worked independently through every step, importing records into EndNote 21, removing duplicates, screening titles and abstracts, and then assessing full texts, with any disagreement resolved through discussion or a 3rd reviewer. To qualify, a study had to be a randomized controlled trial in patients clinically diagnosed with Parkinson's, using FMT with any preparation, route, or dosing, compared against placebo, sham treatment, or conventional care, and reporting at least one motor, non-motor, or cognitive outcome using a validated scale. Risk of bias in each trial was assessed with the Cochrane tool, looking at the randomization process, deviations from the intended intervention, missing outcome data, how outcomes were measured, and whether results were selectively reported. The certainty of every pooled result was then graded using GRADE, an internationally recognized framework that weighs risk of bias, consistency across studies, how generalizable the findings are, statistical precision, and the risk of publication bias. On that last point, the authors are upfront about a real limitation. With fewer than 10 studies feeding into any single outcome, the usual tool for detecting publication bias, the funnel plot, simply isn't reliable here. Trials with disappointing results could exist without ever having been published. Statistically, continuous outcomes were pooled as mean differences with 95% confidence intervals using RevMan 5. 4. 1. Switching between fixed effect and random effects models depending on how much heterogeneity, measured with I-squared, showed up between trials. When a trial didn't report the exact numbers needed, missing standard deviations were estimated assuming a correlation of 0. 5. And 2 of the 5 trials required converting reported medians into means using a standard statistical method, an extra step that adds a layer of uncertainty worth flagging. So how many studies actually made it in? The initial search turned up 109 articles on the topic. After removing 40 duplicate records, 69 unique studies were left for the reviewers to screen. Screening titles and abstracts ruled out 56 more for not meeting the criteria, leaving just 13 studies for a full, careful read from start to finish. In the end, only 5 randomized controlled trials met every requirement to enter the final analysis, and 4 of those 5 used frozen fecal material as the transplant source. Only 1, published by Wang's team in 2000, the delivery route varied a lot too. 2 trials used colonoscopy, 1 used a nasojejunal tube threaded through the nose, and 2 used simple oral capsules. Dosing varied as well. 3 trials gave the transplant as a single dose, while the other 2 repeated it several times over the course of the study. That mix of very different protocols turns out to be one of the central problems running through this entire meta-analysis, and it's worth keeping in mind for everything that follows. Altogether, the 5 trials enrolled 226 participants. 119 received the actual fecal transplant, and 107 received a placebo or standard conventional care instead, without knowing which group they were in. They were conducted in Belgium, in Finland, and 3 of them in China, published between 2023 and 2025. Average age for looking at any results, the reviewers rated the methodological quality of each trial. One of the 5 carried a high risk of bias, mainly because it wasn't clear whether participants and staff had been properly blinded to who got the real treatment. The remaining trials showed milder concerns in similar areas, mostly around blinding and how the treatment allocation was concealed. To actually measure whether FMT helped, the trials relied on internationally standardized scales, starting with the MDS-UPTers, the Movement Disorder Society's Unified Parkinson's Disease Rating Scale. That scale is split into parts. Part 1 covers non-motor experiences of daily living, things like mood and sleep. Part 2 covers how motor symptoms affect everyday activities. And Part 3 is the hands-on motor examination itself, scoring tremor, rigidity, and slowness of movement. On top of that, 2 short cognitive screening tools were used worldwide for this kind of research: the Montreal Cognitive Assessment, or MoCA, and the Minimental State Examination, or MMSE. Let's start with the motor exam, Part 3, the outcome most directly tied to classic Parkinson's symptoms. At week 4, the transplant group scored 42 points better than the control group on average, but the difference wasn't statistically significant. At week 12, the gap widened slightly to 79 points, but the confidence interval was so wide, and the variability between trials so high, with an I-squared around 62%, that it still couldn't be called a real, reliable improvement. And by week 24, the direction had actually flipped, with a small edge now favoring the control group. Across all 3 follow-up points, none showed a statistically solid, consistent motor benefit from FMT. Something more interesting showed up in Part 2, the score for how symptoms affect daily living. At week 12, the transplant group improved by 83 points more than the control group. The result landed right at the edge of statistical significance, with a p-value of exactly 0.05, the conventional line researchers use to decide whether a result is likely more than chance. The study authors themselves describe it as a finding that invites cautious optimism, not proof that the treatment works, but a signal worth chasing in larger trials. By week 24, though, that small positive signal in daily living scores had faded away completely. For non-motor symptoms, Part 1 of the scale, there was no significant difference between groups at either the 12-week or 24-week mark. And on the cognitive side, neither the MoCA nor the MMSE, both measured at week 4, showed any significant improvement in the transplant group compared with controls. Curious about why the motor results at week 12 varied so much from trial to trial, the researchers ran what's called a sensitivity analysis, systematically removing 1 study at a time to see what changed. That analysis pointed squarely at Wang's trial as the main source of the inconsistency, the same one that had used fresh fecal material instead of frozen. That's a meaningful clue. It suggests the viability of the donor's bacteria, not just the fact of doing a transplant at all, might matter for whether the treatment actually has any effect. It's a hypothesis worth testing directly, not a settled conclusion. Make sure the missing data assumptions weren't quietly driving the results. The team reran the numbers under different plausible scenarios, and the overall conclusion held steady regardless of which assumption they used. Evidence that the null result isn't just a calculation artifact. One thing was consistent across every single trial, without a single exception: FMT was safe. The authors explicitly highlight the procedure's good safety profile in these Parkinson's patients. So why didn't it show a clear benefit? The authors point to several concrete, overlapping reasons. The first is simple statistical power. Only 226 people total, spread thin across 5 separate trials, isn't much to detect a real effect, if one genuinely exists. The second is protocol heterogeneity. Fresh material versus frozen, 3 different delivery routes, single versus repeated dosing. Comparing outcomes across trials, that different is a bit like comparing apples that were grown, picked, and stored in completely different ways. Take Bruggeman's trial in Belgium. Frozen stool, delivered once through a nasovaginal tube in 22 patients against 24 controls. Shepherdjan's trial in Finland also used frozen material, but delivered by colonoscopy instead, in 30 patients against 15 controls. 2 Chinese trials, run by Liu and by Cheng, both chose oral capsules with repeated dosing over time, a route that's far less invasive for the patient than colonoscopy or a nasal tube. A third factor is simply time. Follow-up windows of 4 to 24 weeks may just be too short to catch changes that, in a slow, progressive disease like Parkinson's, can take much longer to become measurable. And a fourth, more technical reason. In 40% of the included trials, researchers had to convert reported medians into means to make the data comparable, adding an extra layer of statistical uncertainty to the pooled result. The authors also note that participants' existing Parkinson's medications, especially levodopa, could have influenced the motor scores and made it even harder to isolate the transplant's own effect. As for cognition, the authors point out that cognitive decline in Parkinson's has a complex, multifactorial origin, tied to Lewy body pathology and dysfunction across several brain circuits, something a microbiota-only intervention likely can't reverse on its own within a few short weeks. It's worth being direct about what this means for anyone living with Parkinson's, or with a family member who is. Fecal microbiota transplantation remains, today, an investigational treatment, not an approved or recommended therapy outside a properly controlled clinical trial. The takeaway from this meta-analysis isn't that fecal transplantation is a failure. It's that science, done properly, doesn't get ahead of the evidence just because a theory sounds promising. What's actually needed now are larger trials, with standardized protocols shared across research centers, more objective digital biomarkers that don't depend so heavily on a single examiner's judgment, and longer follow-up periods. Only trials like that will be able to say, with real confidence, whether this therapy, still in a very early stage today, has an actual place in Parkinson's treatment down the line. This isn't an isolated case either. Scientific interest in gut microbiota's role in other neurodegenerative diseases, Alzheimer's among them, has grown a lot in recent years, with results that remain just as preliminary and inconclusive so far. It's important not to mistake media excitement for solid evidence. A biological mechanism being plausible, even promising in animal studies, doesn't automatically mean it works the same way in people, or that it's ready to use outside a controlled clinical trial. If you or someone close to you has Parkinson's and this story sparks interest, the sensible move is to bring it up directly with a neurologist and ask about any registered, active clinical trial that might be worth joining, rather than seeking out unregulated procedures independently. And there's something genuinely encouraging in all of this, beyond the specific result. A serious, transparent, properly registered meta-analysis that's willing to say clearly, this doesn't work yet, is exactly how medical science is supposed to move forward. Source: Liangzhu Mao Ziyan Liang. Systematic Review and Meta-Analysis published in Revista de Neurologia, 2026, Open Access, according to PubMed, registered in Prospero under CRD 420,251,121,443. More health science explained at nexoscience.com.